Good morning, everybody. My name is Jeff Johnson from Nested Knowledge. I'm going to be your host today, and I'm joined by Jade Thurnham and Kevin Kallmes. We are actually doing this webinar at the request of a global center for evidence synthesis in India, and I want to just throw their information up on the screen, give them a little bit of a plug because without them, this webinar would not be happening today. So, yeah, check that out on your screen. You can. I'm going to be throwing up links as QR codes today. So if you want to follow them, you'll have to pull out your smartphone and you'll have to scan them and that sort of thing. Because it's a video, right? So I'll try to include all the links that I'm throwing up on the screen today in the description of the video if you're following on after the fact and you don't have a QR code reader. But hopefully that gives you a sense of where to go and how to follow along while we are presenting today. So yeah, let me just throw it to Jade and Kevin. Jade first. Do you want to just talk a little bit about who you are and what you do at NK?
Yes. Yeah. Thank you, Jeff. Hi, everyone. I'm Jade Thurnham. I'm the Director of User Experience and Design at Nested Knowledge. So I work on a little bit of the design side. I work with some users. I like to come on and do these demos. I work with academics. Lots of the good stuff that you tend to have in a startup. Yep, that's a little bit about me. I'll throw it straight to Kevin.
Thanks so much. I'm Kevin Kallmes. I'm co-founder and CEO of Nested Knowledge. Before Nested Knowledge, I focused heavily on evidence synthesis as a research practice, where I was doing it in Microsoft Excel. So a lot of what we're seeing today, not only out of NK, but I think just general advancement in research practices, it's really exciting. And so I'm also excited to be able to present at least what Nested Knowledge can contribute to best practices in evidence synthesis.
Phenomenal. I also just want to highlight as well that you can, as a viewer today, comment or ask questions in the chat of whatever platform you are watching on today. So whether it's LinkedIn or YouTube or Twitter, you can actually just comment into that little box down there and ask us questions live. And I'm going to pluck out some of the best ones, and we will answer them live. Of course, if you have questions in the future after this live stream, finishes, feel free to just hop over to support at nestedknowledge.com, and you can email support at nested-knowledge.com, or you can just follow that little QR code right up there at the top, and that's going to take you to our support page. We have all kinds of great resources, including the email I just mentioned. And the ability to ask for new features for us to build into our software, or just read helpful support articles and that sort of thing. So I just wanted to throw that out there up on the screen just so you have it as we go today. So I want to first turn to Jade to talk a little bit more about what evidence synthesis is and how Nested Knowledge can tend to help you with that, speed along the process, make your life a whole lot easier while you are synthesizing evidence of all different types. So yeah, Jade, go ahead, take it away.
Cool. Thanks, Jeff. So thank you everyone again for joining us for our session on evidence synthesis. Today, we are partnering with the Global Center for Evidence Synthesis, as Jeff mentioned, a fantastic nonprofit organization, helps with research, education, and training. They're dedicated to gathering comprehensive and unbiased syntheses and have lots of resources on their website, which you can scan the QR code, take a look yourself to conduct your reviews. But in particular, today we're going to discuss Nested Knowledge, a platform to conduct evidence synthesis all in one place. But first, what is evidence synthesis? So as most of you may know, evidence synthesis is the process of reviewing all available research and performing analyses on a particular topic. This can be any type of review, systematic or otherwise, rapid review, meta-analyses, etc. But what exactly is the point of performing these reviews? Well, the process of evidence synthesis impacts so many areas of our lives. In healthcare alone, it determines what disease treatments are available, what drugs are on the market, it affects the production of medical devices, healthcare policies, all types of evidence-based practice that we engage in. But the issue is, currently there is no one space to perform these reviews and synthesize evidence. Current processes have us filling out different spreadsheets, downloading and keeping track of all these different files in all these different folders, which is just a huge pain. So what if you could do it all in one place? Nested Knowledge is a web-based software that is the first to allow you to conduct your review all in one place, with each step assisted by artificial intelligence. You can conduct your search of up-to-date research papers, screen each of them, extract data that is specific to your project, perform critical appraisal, and all while interactive visuals of your data are automatically generated in the background. And today, we have Kevin, our CEO, who is a serial entrepreneur and an expert in the field of medical devices and research. He will be doing a demo of the software to give you an overview of our features. And as Jeff mentioned, please continue to submit any questions through whatever channel that you're watching us on, and we'll do our best to answer them all. We hope you find this demonstration of nested knowledge helpful, for evidence synthesis helpful, and that's all from me. I'll hand it over to you, Kevin.
Hold on. Speaking of questions, we've already got one in the chat here. So Kevin, I'm just going to toss this one right to you because I think it's an excellent one. It's really topical here. So Muhammad asks, do you think the process from including a study to extracting data for that study made simpler and involved less clicks, setting up tags, preparing hierarchy, etc.? I'm noticing some nested knowledge specific language in there. So Kevin, you'll have to maybe elucidate the process for those of us who are just finding out about nested knowledge today, and that sort of thing. And so yeah, actually, yeah. So I want to give you a chance to hold that to take that question, and then Kevin, before you dive into yours, I am actually going to pull in a few of our friends from GCES, and they can maybe plug themselves too. So go ahead, answer that question as quickly as you can, and then we'll hop in and pull them in.
Okay, great. I'll hold off on screen sharing and demoing for a second, but I think that's an amazing question. It shows an understanding of the process, and so I think it's a good question for me to generalize and say, okay, evidence synthesis is trying to do the things that Jade's talking about, but how do you do it? And usually, to me, I would deconstruct it into the simple steps of you're going to search or find records, you're going to filter them, including and excluding records, and then you're going to extract data. And in those three general steps, you're going to get from I have a research question to I have a data set. And often you'll also analyze that data set inside or outside of nested knowledge. And I will actually say that this is one of the most underestimated ways to improve the process. We all know that evidence synthesis is incredibly work intensive. The average literature review, according to a literature review on the topic, I found was about 18 months. And I thought that was wasteful. Not that people are doing their research incorrectly, just that they are spending too many times quite literally clicking in Microsoft Excel, or they're tracking themselves. They have to both include the study and do all the administrative background work in order to follow their own research. And so one of the major goals in designing nested knowledge is reducing administrative burden of a review. And so going from the process of include a study to extract data for that study with as few clicks as possible. I mean, Jeff is our chief designer, and I think that he can tell you exactly how many discussions we've had about going from 2 clicks to 1 click for the include process was a big thing for us. But if you generalize that out, you can just say, we want to make a streamlined process where you're going from not only include a study, but find a study in the first place through to have fully extracted data in an interactive visual with as few clicks as possible. That's the goal of the software. That's the goal of what we're going to be demoing today. And I think that is, I think that making that simpler and easier is an additional benefit on top of all of the interactive visuals, AI assistance. It's a sneaky benefit that you get. But
absolutely, though I'd be the first to admit too that we are still looking for ways to make that process even faster. Of course, it's a process that's always going to require humans, and we can talk a little bit about how we're trying to augment that process, take even more clicks away from your process with things like artificial intelligence. But ultimately, it's still always going to be a manual process. There's so much creativity in the evidence synthesis process, and we're going to get into all of that today. I want to just turn to the GCS folks and let them, GCES folks, I should say, and let them kind of talk a little bit about their organization and what they do over there. So teach me how to say your name. Is it Dr. Bajay? I'm gonna butcher it.
Hi, Jeff. Am I audible? Yeah. So
Okay.
my camera, this system, my camera is not working. So
That's okay.
my apologies. Thank you very first. I thank everybody, Jeff, Jade, and sorry, I forget the name.
No, you're good. Jade, Kevin, and Jeff.
Yep.
Kelvin. Okay, so accepting our request to spend some time with our members. I hope there are, I do not see how many members have joined right now because this system is not allowing us. It's good. The organization we started recently, maybe it is only 6 months of old organization, but within 6 months we have more than 1,000 members joined. We basically, the organization started at PGI, which is a tertiary care hospital in India. It's the second most highest ranking hospital in the country after the All India Institute of Medical Science, New Delhi. We have many domain specialist people are there in members, starting from all medical domains, also from social science background. We do lots of evidence synthesis work. If you can visit our website, we have so many publications, starting from systematic review, scoping review, meta-analysis. So I just recently find your website, and since we are trying many applications, and I find this is one of the, I just named it one-stop solution in evidence synthesis. So which probably that we are looking for, and hope to learn, our members will learn something more today's session. So Dr. Arvind, do you want to say something?
Thank you, sir, and good evening all. Myself, Dr. Arvind, and I'm working with Dr. Bijaya in GCSF for the last 6 months. And I totally agree with whatever sir has mentioned so far. I find it interesting actually regarding a one-stop solution, since we are using multiple applications for each step, right from the time of finding out the research question, then importing the potential studies, then screening, then data extraction, then writing. So all these things are very intriguing to find all these in a single place, and I would like to know more about it. And I also have a couple of queries which I might be asking at the end of the session. So if it is answered during the session, they won't be necessary. But then let me keep it for the end. So thank you.
Thank you. That's great. Thank you for the introduction to both of you. Kevin, I'm going to toss it back to you. Let's talk a little bit about our software, and let's just show people. I think that's often the easiest way to just dive right in, right?
Absolutely. And I just want to say thanks so much for putting this together. This was all at the initiative of GCES, and I know what it's like to start an organization from scratch. It sounds like you guys have gotten off of the ground a lot quicker than most young organizations. So I'm glad that you're already able to jump in and do research rather than having to do some sort of scaling activities in your first 6 months. It sounds like you guys were able to jump right in. But let me jump over to our software. And I'm going to start not with a demo of the exact functionality, but with a description that will give you at the highest level how the software works. And when we say a one-stop solution, we actually mean not just for completing your review, but also for examining, analyzing, and presenting your data. So we actually, at Nested Knowledge, split our platform into Autolit, where you're going to go through those steps that I mentioned just earlier, searching and finding studies, screening them to determine which are relevant, and then extracting data from them. And all of that is going to automatically create your interactive, living data visualizations in the synthesis page. And so to drill down even a little bit further, when I say search, I don't just mean Googling for articles. I mean following best practices for systematic review, where you're going to create as comprehensive as possible of a search strategy. We have an AI that assists you in doing so. And then you can run that search not just externally and then import a file, but you can actually run searches directly on Nested Knowledge to pull records in, both those that are published today, and you can even set a repeat cycle for records that are going to be published in the future and could be included in your updatable review. Then at the screening process, and Jeff, please do interrupt me if we're getting questions, by the way,
will do.
in the chat that are related to these things. But yes, then after you've run your searches and you've set up that repeat cycle, you also can use AI to assist with your screening process, where you come into our software and in one click, you're saying whether studies are in or out, you're including, you're excluding, and giving a rationale. And our AI learns from every single decision that you make. So you start a project, you start screening studies, and 50 studies in, it should have a pretty good idea what sort of research you're looking for. And it's going to give a score to every underlying study. And then that's going to take half of those clicks off of your plate in the actual screening process. Then, and this is one of the things that we're the most proud of, that we think is a distinguisher of nested knowledge, is using ontologies or customizable tagging hierarchies to extract the underlying concepts from the literature. And this is an incredibly flexible capability. Anything from study methodology, setting, all the way through to outcomes from the patient population, all of those can be extracted into tagging hierarchies that you can use and reuse, and that are a very good way to both transparently reflect your research question and what you're trying to gather, but also it sets up our interactive data visualizations. And this is actually one that is embedded in our About page. This is an interactive qualitative synthesis diagram. You can see here a hierarchy, an ontology of population characteristics, of outcomes, and of study characteristics from the treatment of acute ischemic stroke. And Jade mentioned that I have a background not only in medical research, but in medical device. Before Nested Knowledge, I actually designed thrombectomy devices, which are methods to pull clots out of your brain. And they're generally compared against medical therapies like thrombolysis. And so you can see here that we actually completed a review comparing thrombectomy devices, the type of device that I used to design, to standard medical therapies with respect to outcomes such as neurological outcome, hemorrhage, mortality, and so on. So this is an interactive diagram where I can drill down on these concepts from the underlying literature. If I click in on any of the articles that I've included in my review, I can get an AI highlighted abstract. I can get all of the data from that underlying study and also all of the tags that informed this diagram. So in effect, the process from search, screen, tag, you can get an interactive systematic review out of that. And then if you want to go further and complete a meta-analysis, we also have automated network meta-analytical statistics built in on top of that. But to show those, I actually want to jump over to the Nested Knowledge native. This is the synthesis page for an acute ischemic stroke review. Again, we're still on the topic of comparing thrombectomy devices to thrombolytic drug therapies for the treatment of stroke. And as you guys saw in the embeddable version of this, you can drill down on not only highest level interventional categories, but specific devices that are used to treat stroke. You can drill down on any outcome that you reported in your systematic review. You can also, and this is something that I find really helpful for communicating. If I'm the researcher and I've been deep in these studies and I want to communicate insights that go beyond just the contents of, or like the exact text contents of any study, I can also draft an insight. So in this case, you can see that thrombectomy, the types of devices I used to design, have been shown in randomized trials to significantly improve neurological outcome. And I can find this in my interactive review simply by clicking on that insight, seeing the text that was composed by the expert, but they've also pre-filtered this diagram. So they've drilled down on the exact concepts, the exact tags in our hierarchy that prove out that claim. And then, of course, I can always go in and check whether that claim is actually correct by reading through the abstract or examining the data myself. In addition to interactive visualization of this point and click systematic review and of insights, I also have the interactive automated network meta-analysis. And so I'm going to jump over from qualitative to quantitative. And if anyone's wondering, how did you get to these diagrams? Don't worry, we will be getting to the entire process. We're going to show the whole one-stop solution, but we wanted to show you guys, what are you building toward in your nested knowledge review that distinguishes it from any other systematic review? But let's jump over to our interactive network meta-analysis. Here you can see basic summary data with automatically calculated 95% confidence intervals across each therapy. So think of this as your spreadsheet. This is where I'm going to examine any of the underlying data. It's much more manipulable than a spreadsheet though, so I can drill down from the highest level, the types of therapies, all the way down to combination therapies. If this were for drugs, I would have doses and dosing schedules and other subgroups of my therapies. And if I click all the way down, I'd get to study level data, for which I can examine the exact data reported from the underlying records. In addition to summary data, we also have the ability to complete network meta-analysis. And just as a a background, I'm sure that many of our viewers here know this, but network meta-analysis is a method for using imputation to compare therapies that might not have had a direct comparison in say an RCT or other comparative study. So here you can see if we break out the therapies for mechanical thrombectomy, stent retrievers and aspiration are two different devices that are used to retrieve clots. We can see that not all of the therapies of interest necessarily have a direct comparison, but we can still use imputation to get a rough odds ratio for a given outcome. So here you can see an odds ratio chart. Each of these is an odds ratio between the therapies on each of these axes with respect to mortality. I can also shift it over and do it for hemorrhage. And for each of these, I'm just going to get a comparison of each of the therapies that report this outcome. And if there's a direct comparison, I'll be able to click in and get a forest plot for those exact comparisons. So for instance, I can click in here and I'm gonna get a forest plot for all of the exact underlying studies that report symptomatic intracranial hemorrhage for mechanical thrombectomy with thrombolysis against thrombolysis alone. I can then use these odds ratios and these forest plots to come to conclusions about likely differences in patient outcomes based on device used or based on therapy used. And if you're wondering, on top of this, well, there are two questions that I often get, so I'll just ask them to myself. What about heterogeneity? So of course, if I have a forest plot that tells me that mortality is reduced in thrombectomy compared to thrombolysis, and a lot of underlying studies that might contribute to this, but I'm uncertain about the data supporting that claim, I can examine the I-squared values, which are generated automatically in the software. But more importantly, I can also drill down on a funnel plot of the exact underlying studies and how they've contributed to this finding. So any study that might have small study effects, any study that might be an outlier or an influencer, so something that has an undue influence on this synthesis of evidence toward this network meta-analysis, I can come in, I can view those studies, and I can click in, examine their details, and see if it's actually because of the therapy or it's because of some background bias. So we always have these tools for examining bias. Even beyond the automated tools for examining bias, like these automatically built funnel plots, we also have critical appraisal tools. You can see here that the Cochrane Risk of Bias 2 survey was completed on top of this acute ischemic stroke systematic review and meta-analysis, which allows me to examine at the highest level what was the level of concern around bias in the randomization process, in missingness of outcome data, and so on across the included studies. The only other things I want to say about these outputs, I should say, on synthesis before we move to the actual process. First, these are embeddable, as you saw on our About page. You can create this embeddable yourself, or if you would rather make your sharing even easier, you can link share these. So each individual page or synthesis as a whole can be embedded. You just come to this upper right-hand corner and you can grab a link for it. You can grab the embed code. This is the same as embedding a YouTube video or similar, except unlike a YouTube video, you'll actually be able to interact with the contents. So if you're looking for ways to share your evidence that go beyond emailing someone an Excel sheet, you can link share your synthesis page or any subpage of synthesis, and you can also embed these pages in another web page. And then the last thing I want to note is that like the question that Mohammed asked at the start, how can we reduce clicks? We don't just think about how to reduce clicks from the inclusion step through to the data extraction step. We also think about how can we reduce your clicks in your administrative tracking of your own work. And so we actually automatically build out your audit record of all studies that come into your review, which records were excluded at which points, for which reasons. And we build out your Prisma chart that is not only reflective of your current state of your nest, but I can also filter this and see the state of our, I didn't even give a definition for the word nest, I'm remiss. A nest is an interactive systematic review completed in nested knowledge. But I can see a Prisma chart that shows, again, the flow of records through my nest for any previous date. So if I wanted to see the state of this nest at the beginning of this year, I can see how many studies had been included, how many studies were awaiting, and how many studies were added by bibliomining or by expert recommendation. All right.
Yes. So Kevin, we've got a couple questions in the chat, and I just want to editorialize here. It's everything you just saw, right? So everything that Kevin just showed you, all of those calculations on the back end are done automatically through the sheer fact of you extracting, screening, tagging, searching for data on our platform, right? So it's just through the process itself. And Kevin's about to show you the autolit side, like how you actually get all of your records into the nest, as we call it. He's going to show you how to do that and what those best practices are, and all of that stuff. Once you are done extracting data within nested knowledge, quantitative synthesis, qualitative synthesis, critical appraisal, Prisma, all of those calculations are done, and that is how we are generating these outputs. So there's no fancy AI necessarily doing all of the work, right? We have this philosophy of AI augmentation, and we're going to show you, exactly which places in our software where that comes into play. But ultimately, it's up to you to decide what your ontology looks like, which interventions, which comparators are of interest to you. Let me hop over to some questions, and then I'll toss them back to Jade and Kevin as it makes sense. So we've got, I'm sorry if I mess up your name here, but it looks like Dr. Naba Sneezy. Okay, so I'm sorry. Is it free to access the Nested Knowledge platform? Jade, go ahead, take that one.
Absolutely. So it's free to create an account. Everybody gets their first nest for free.
Right. So everyone gets their first nest for free. After that, we have various pricing options because, of course, we are a for-profit business. So after you have completed one entire systematic review, everything you saw, you can do that for free right now, today, in our platform. In fact, let me just toss up the QR code and allow you to sign up here. You can just scan that QR code and fill out a few details about yourself. We just ask for a name and an email address, and you can sign up entirely for free and start doing all of this stuff. After that, it's $20 a month for academics. However, if you are looking for some sort of discount, feel free to email sales@nestedknowledge.com. You may have certain extenuating circumstances which entitle you to that discount. So feel free to inquire. Otherwise, it's just $20 per month per person to use our platform after that first free review. So check that out. Other questions?
Sorry to interrupt you, Jess.
Yeah.
I think it's just good to emphasize that we do want you to try out the software, try it for yourself, see if you like it, and then consider the pricing options. But I think it's most important to us for you to see that this is a powerful software, that it works, and you're always more than welcome to ask for support too, through our support channels, through our email, and hopping on calls like this.
Right. Yeah, I'm gonna toss that email, the best way to contact us in the up on the screen in just a moment. I want to get to a few more questions here as well. Same person asks, is it help you to extract the data? In other words, does it help you to extract the data, or how do you import references from into Nested Knowledge? That might be a good segue. I will throw up that contact email just so you can see it in a second. But Kevin, why don't you just go ahead and share kind of how the autolit portion works? So how do you get data into NK?
That's a great question. And I want to cover two basic background concepts before I start jumping in on individual features. But let me do that a little bit slower. We're now jumping from the synthesis page into the backend. So from your outputs that you're going to share with your readers to where you actually complete your review. And a couple of things about our process and the question of, do we effectively extract the data for you, or do we help you extract the data? There's no step in the autolit portion of the software, no step in the systematic review process that we fully automate. And this is a philosophical decision around understanding people's concerns about accuracy and people's concerns about who is deciding. And so when it comes to including AI assistance, as you're actually about to see for our search feature, we take the stance that you should be defining the final inputs into your project, and we should be assisting you in getting there with AI. This is because we've heard from many researchers, from academics, from regulatory bodies, from uses in health economics, all of them have told us, be very cautious about using AI in medicine. And we've seen many things come through recently about large LLM models being used for, writing manuscripts, being used for a lot of scientific tasks. We're excited about the use of AI, but we also want to be careful. We want to make sure that we're only launching AIs that are going to be assistive and are going to be accurate. And you end up being I like to think about it as like a partnership between the human side of the software and the AI side, where you are going to give your inputs. These are the concepts I care about. The AI is going to run out and try to find other studies that are going to be relevant to your topic. Then at the screening stage, you're going to teach it what studies are relevant to you, and it's going to start scoring those studies, but always with a human giving the last decision on whether a record should be included. And then at the extraction stage, we're going to find the data within the PDF for you, but you will have to extract it, because any direct AI extraction would be a potential risk that an AI is bringing an inaccurate result into a systematic review, which, again, we've been told over and over again, we should be philosophically very, very careful about launching an AI that could cause any inaccuracies or could effectively be making a decision about an inclusion or about an extraction in the place of a human. So I know that's, I don't want to stay too long on that point, but you will see it in every feature throughout this: AI assistant, and then a human completing the review task. But with that, I'm just going to jump straight into our search. So as you saw at the top, every systematic review should start with a research question that you're going to turn into a search strategy. And I'm not a medical librarian. I've worked with some wonderful medical librarians who have a myriad of methods for finding or for building the search strategies that will help you find records on multiple databases. But lacking that experience myself, I like our search exploration tool as an assistive to help me build out my PICOS, which is my population, my intervention, and my outcome, population intervention comparator and outcomes for a given project. And then I actually want to build out that PICOS strategy into the set of terms that I'm searching. And this one started with just typing in the word stroke, because that was the concept we were searching, adding it to our preliminary search, and then grouping those terms by like concepts. So you can see here, we're grouping together population terms, we're grouping together different interventions for stroke, we're grouping together the outcomes that we want to collect. But I didn't come up with all of those myself. And by the way, if I'm changing anything about the structure of this, you can see that it's live building out my Boolean query on the right. If you want to talk about removing clicks, what about removing keystrokes of typing and or and parenthesis and figuring out how I've screwed up my structure here? All you have to do is put in your term, and then we will build out your Boolean search for you. So that's yet another time savings piece there. But the strategy is really the important thing here, figuring out the PICOS, and then figuring out not just the highest level stroke. What are synonyms for stroke? What studies will I miss if I don't add either synonyms or like terms to stroke into my search strategy? There could be studies that do not mention my exact indication. There could be studies that do not mention my exact interventions. And so the AI is going to recommend across all the abstracts that come back from my preliminary search, it's going to identify population terms, intervention terms, and outcome terms that are commonly used. And if I click in on these, I can also get mesh definitions that will be automatically searched if I select these. So in effect, you can think about this AI, this RoboPico, as your search strategy assistant, where you start with just a couple of words that identify your topic, and then it builds out the synonyms, the related concepts, and things like the outcomes that you might want to extract. This also gives you a high-level view. What does the literature within my current search report? Well, it seems like it's reporting a lot of things about stroke, atrial fibrillation, about your circulatory system, about large vessel occlusion is a specific type of stroke. And then for therapies, we're seeing a lot of different drugs that are for anticoagulation, for removing clots, for basically stroke treatments at the highest level. So thrombolysis is the general term, and then apixaban is a specific drug. All of these are potential search terms that I should be adding to my search before I move on to the screening stage. But when I'm done building out my preliminary search strategy, I can just hit finalize in the upper right-hand corner. And as I said, we are able to run searches directly on PubMed, on clinicaltrials.gov, on Europe PMC, and also open access journals. And I'm also able to schedule this search. So I can just take this search that I've built in the search exploration with AI assistance. I can schedule it daily. I can run it on PubMed. And if I hit add, it's going to run this search, and it's going to rerun it every day and queue up the studies that return from it. Additionally, if I've run searches previously, so if I have a spreadsheet of studies from a previous review, if I have a search that I've done on Embase, I can also trivially import them here. All I need to do is come in, go to File, Import, enter the query that I used for tracking purposes, right? We should have a highly auditable, highly transparent review, and I'll be able to import that bibliographic file or that spreadsheet. So I can always have my living searches running alongside imports that I complete myself. A couple of other extras, and these are not necessary, but just things that I think might be helpful. We consider ourselves data source agnostic in the sense that if you have a source that is not a published study, you can either bring it in manually or add it as a PDF. So if you have a PDF of, say, a real world evidence study or internal data that is not yet published, you can simply upload it. And we have an AI that'll pull the metadata and add it right into your project. Also, if you have an existing systematic review that you've already completed and you just want to pull the references out of it, we also have a feature where you can bibliomine in one click. So I upload my existing systematic review as a PDF, and the AI goes in, it goes into the reference section, and it rips out all of those citations and adds them to my project. So you can build upon the work of others. And I think this is an important piece about replicability. If you're completing a systematic review on a topic and you find someone who has done one previously on a similar or related topic, it's very important that you're going to capture as comprehensive as possible of a set of studies. So you should be able to, or it's advisable to use their reference list as a way to ensure that your search strategy is complete by supplementing it with other people's previous systematic review work. But again, so at the highest level, we use AI to assist in the search exploration and search strategy. We then run those searches either live on PubMed, or we can use import from databases like Embase. And then we can also add creative source types and complete bibliomining. So lots of ways to get a record into your nest, and that will all be funneled into your screening. At that point, we can move forward and simply examine abstracts. And as we said, we try to eliminate as many clicks as possible. Here we are in what we call two-pass dual screening, meaning that for every record, we're going to have two people review it at the abstract level and either advance it to full text screening or exclude it. Then we're also going to have a full text screening level where everything that's advanced is examined again by two people. And at both of those stages, we're going to have a third-party adjudicator who can examine all studies where the two people disagree. So my screening process will be as simple as examining this record that has come back from our search and advancing it or excluding it as not an RCT. And as soon as I hit not an RCT, I will get the next record. And that record that I've just screened will go forward to the adjudicator. Usually someone who's an administrator or a senior member of the project is going to examine my screening decision and then the screening decision of the other human. But there's more here. And the AI assistance here is quite subtle. So not only do we have the AI highlights, so the same AI that helps with search strategy also highlights those population, those intervention and outcome terms. I can also add my own personal keywords and I can color code them, or I should say actually my nest specific keywords. These are shared with your team. But in addition to that, we have an AI that we call Robot Screener. And Robot Screener, as I said in the About page, is an AI that learns from every inclusion or exclusion that you complete, and then it builds a model where it compares all of your studies that were included or advanced against those that were excluded. And then to every study that is still awaiting your screening, it's going to assign a score. Then, if you want to turn on Robot Screener, you're actually going to be able to replace one of your human screeners with that AI. So instead of it being, say, Jeff screens, Jade screens, and then I adjudicate, it would be Jeff screens, an AI screens, and then I adjudicate. So there's still a human giving that last decision, but the AI is saving half of your upfront screening work. And we also are big fans of transparent AI. So this is showing all of the predictions across included, across excluded, and unscreened studies. And it also gives its accuracy. So you can see here we have an area under the curve. We have the recall score, precision, F1. We want to tell you how the AI is doing in your specific nest, so you can choose whether or not to employ that robot screener. And in general, we find the more studies you screen, the more accurate the robot tends to get. Obviously, different research questions might have complex inclusions, like maybe you're including a vast range of studies. For this one, since it's RCTs only, there's going to be a lot of consistent trends that the AI is going to be able to catch on to pretty quickly. So you can employ this after, say, 50 records. But it is nest specific, which is why we give these these transparent accuracy scores on the AI. But that means that instead of needing multiple people to screen these studies, I can just include or exclude, and then it will go straight forward to the adjudication without another human. All right, so let's assume that we have now advanced studies to full text screening, and then for those full text screenings, we have employed a human and an AI and then an adjudicator to determine which studies should be included. And the adjudication is as easy as picking the reason. So here you can see that the robot excluded this, and so did we. I can also auto-adjudicate any agreements. So anytime that a human and the AI agree, I don't need to examine it. But let's assume that we've finished with this process. That's where we would move forward to building out our tagging hierarchy. But I do want to take a pause here because we're moving pretty quickly through these features. And Jeff, I want to throw it to you for questions.
I was about to say, we've got some really good questions in the chat, and I want to get to as many of them as we can. Nicole Fusco, I think is her name, asks, can you upload a whole search, for example, from Embase, which requires licensing? I'm going to toss this to Kevin. Go ahead.
Yes. So if you are using Embase and you want to upload records to Nested Knowledge, the method you should use is the RIS export. So in your under your Embase subscription, you should be able to run your search, and at the end of that search, you will have the option to export. Use a bibliographic file like RIS as your export, and that will be directly importable. You go to Lit Search, you go to Add and File Import. Here, you can just upload that RIS file, and then that will pull in the metadata from your Embase search and add it into the nest. It will also automatically deduplicate it against any other searches. So if you have a PubMed search and you're importing that Embase search, it's going to compare all the metadata and it's going to make sure there's only one version of every study. Great question.
Next one, I'm going to toss this one to you as well, Kevin. Then I got one for Jade. Amit asks, is it possible to exclude the studies in data extraction part, which I had included during the screening process? And I was going to say, you got to show off Study Inspector. A lot of people sleep on Study Inspector. They don't see it right away.
So this is something I often say for the end of demos because it is our most expert level feature. It's something that I think takes some learning, but it's incredibly powerful once you get there. So Study Inspector is your library of every single study that you searched or that was ever brought into this nest. So every single study that's been in this nest, I can view from here, and I can do any step in the software. So I can do abstract screening, I can do full text screening, I can tag studies, I can meta-analytically extract them, and I can even complete my critical appraisal survey, all on this study, all from this single view. So I'm able to filter. So let's say that I wanted to filter to all included studies and re-examine them and make sure they were in fact included correctly as was it directly asked. Let me say that I'm at the tagging stage and I've already been tagging this full text. I think that this one is exactly what we're looking for until I get to, say, a methods section where it says, oh, this wasn't randomized. And I realize that we should have excluded it earlier. No worries. I come to Study Inspector. I jump into full text screening. I adjudicate this out. So I can say that this is not an RCT and I can adjudicate this out. And once I've done so, that will take it out of the tagging queue, and it will also take it out of the synthesis visuals. So let me generalize this. Any screening decision, any tags that you applied, any extractions, critical appraisal, any step you've completed in the software, if you need to revise it, use the filters and then click in on the relevant study in Study Inspector. Even beyond that, and I don't usually demo this in the first presentation, but let's say that you systematically think that there might have been some errors in your screening, and we filter. Like, let's say that all studies that were tagged with thrombolysis, I actually want to exclude. And this could be for any reason. Let's say that I wanted to make a copy of this nest, spin it off, and do an RCT of all studies that don't report thrombolysis. I can come in here and I can use a bulk action to update the screening status of these studies. I can say, I want the final full text decisions to be exclude. And I can change the inclusion status of hundreds of studies at a time in just a couple of clicks. I can say the reason, and then I hit apply. And once I hit apply, it will in bulk apply that decision to all studies filtered in Study Inspector. So you can probably see where this gets quite powerful. I can apply, as long as I apply intelligent filters, and we have a lot of different filters on bibliographic data, on where the study is in the workflow. So like, is it finished with tagging? Does it have a full text attached? And you can even filter by the tags you've applied to studies. And then once you've applied those filters, you can in bulk make changes to the contents of your review. So that is a little bit dangerous in the sense that you can very rapidly accidentally exclude dozens of studies that you didn't want to. But if you have a clever filter and you know what you're trying to include, exclude, tag, or so on, you can use bulk actions to do that across multiple studies at once. Probably more than was asked there, but I hope that was helpful. Jeff, back to you.
Flexible structure. It's a hallmark of the NK platform. Okay, Jade, this question comes to us from Malia. Does someone need to have a subscription/account with Nested Knowledge to view interactive review with the generated link? I believe this is a question related to synthesis more than anything else.
Yeah, great question. So in order to view the synthesis, which is the interactive visuals, you don't need an account or a subscription. But that is provided that the nest itself, so the review, was made public. So anybody can review it provided that the nest was made public. Yeah, you can go into settings and toggle with the nest access. But in order to view the autolit, so the workflow portion that Kevin's going through, you do need an account and you also need access to the nest. You need to be one of the collaborators that have access to the nest. But this can all be toggled in the settings page that Kevin's showing.
Right. And I think, Kevin, you could maybe talk a little bit about this too, but I think that's because we want our platform to be amenable to a variety of different use cases. So we work with, a lot of folks in industry where privacy is of, paramount importance, where you need to keep your data kind of in-house and proprietary, or at least only share it with the stakeholders of your particular project, So I'm thinking specifically of like HUR folks who are working within a CRO type environment, a contract research organization, and then they are working on, a meta-analysis, for example, for a particular client, right? It's a particular pharmaceutical company, and they don't want anyone else to see their project. That's possible with this setting and this platform. Similarly, it's possible on the academic side to publish these outputs, and you can have truly living systematic reviews live on the internet that are updated on a periodic schedule by the researchers in the back end. So journals, societies get really excited about our tool as well, because they can stick that sunburst diagram side by side with traditional text outputs on the internet anywhere. And as long as it's public, everyone's going to be able to see that and interact with the underlying data. And because of the way that we built this platform, you don't have to worry about things like, things like copyright, because we're not showing the full text. We're showing extracted data. It's part of a visual. It's no different than including a diagram, except that this diagram is interactive and it's living. In other words, it's updated on the back end by researchers. And that's kind of a unique offering of Nested Knowledge. Okay, Kevin, let's dive right back in. Let's talk a little bit about ontologies, flexible ontologies. Maybe you could just start by addressing how this is different from some other ontologies that people might be familiar with, namely that it's flexible, that you determine what's important to you when you are generating this ontology.
Absolutely. And these are all great questions, but I'm just going to jump straight in on tagging hierarchies. The difference between this and, say, Mesh. Mesh is a hierarchy that's created by the great librarian team at the NLM, and they are covering every topic under the sun within medicine, right? They're going to cover different therapies, they're going to cover different drugs, but it's going to be at the highest level, and they're using those to tag studies just to help find them. Our tagging hierarchies are both meant for tagging individual studies. So you're not just doing this in general and saying, like, let me do a search for studies with this tag. You're finding the exact text fragment from the underlying study that proves that a given outcome is reported within it. But more than that, you're actually building these hierarchies yourself. You don't have, or you don't need to depend on the Mesh hierarchy for what goes into your nest. Instead, you should be reflecting your research design in your hierarchy. What study methods or study details do you need in order to understand your systematic review? What patient characteristics might be impactful on the outcomes of the interventions that you're collecting? What are the interventions? What are the subgroups that might be reporting different sets of data that need to be compared in our NMA? All of that is built out here, and you can probably see both the qualitative synthesis and the interactive network meta-analysis depend on our tagging hierarchy. So we have to be very careful in how we build out what interventions we want to compare, because they need to match not only what we want to compare for our research question, but also they need to be reported in the underlying studies. So it has to be both reactive to the contents of your review, and it also has to reflect what you're trying to do. These can be built as simply as creating new tags, and saying where they should live. So if I have a new study detail, I can simply create it, name it, and it'll pop up into my hierarchy. I also can import past hierarchies. And this one is actually based on a stroke starter nest that I built out. So I didn't have to build this when I came in to create this nest. I was able to grab this hierarchy template, and then I was able to import it and start from there. I can then customize it to my specific research question, but that saves a ton of time. Jeff was talking about different organizations, and we want to be very supportive of organizations providing intellectual capital, let's call it, providing these background structures that make research easier for the members of their teams. So you can use organizational templates to build out your hierarchy, customize it then to your exact use to answer your exact research question, and then we're going to jump straight in on underlying studies and be able to tag them. So I can simply scroll through here and find underlying tags, or I can use AI recommendations. And this is where that assistance comes in. If I don't want to read this full 3,500 word PDF, I simply click on the tag recommendation. It will scroll me to where the data are reported. I can either simply apply that tag, or I can find the text fragment that I think proves out the statement that I'm going to make. I then grab that text fragment and I can hit apply. And as soon as I do, it will save that text fragment. It will present it in qualitative synthesis, and it'll allow me to auto scroll back to it at any point. This will also be assistive at the next step where I can complete meta-analytical extraction. That is optional. There's no requirement that I complete this meta-analytical extraction. But if I jump forward, again, let's go back to Study Inspector and use that functionality to go to the next step. Once I've tagged that study with the AI recommended aspect score, I'm going to be able to jump in on the extraction phase, and anything that I have tagged will be saved and I can jump to it. So if I want to extract mortality, I can just filter to mortality. I can click on the tag and it'll jump me to where mortality is reported. I can then extract it, and that will be put into my automated network meta-analysis. So the extraction process can basically look like take the AI recommendation on where to tag, go to the extraction step, click on the little tag icon next to my data element. It will auto scroll me to where something is reported. I need to extract the time point, the data, and the exact event rate in this case. If it's say age or like mean age, I would extract the mean and the standard deviation. If it was median, I would extract the median and the IQR. If it's an event rate, as for mortality, I just need to extract two numbers for the two different therapies, and then that will be fed automatically into our network meta-analysis. And that is the entire process for extracting data into nested knowledge. I know we've gone a little bit quickly through it, but if you search, get your records in. Screen, decide which ones are relevant. Tag, identify where those concepts are reported in the underlying study, and then extract any quantitative data I want to meta-analyze. I now have this living, interactive, systematic review and meta-analysis that I can present in synthesis, and that I can also then use for my more conventional outputs. And I do want to come back around because we're coming to the close here and say that if you are concerned about, I don't want my systematic review to only be in this qualitative synthesis diagram. What if, I need to run other analyses on top of it? What if I need to send someone a spreadsheet of my data? Never fear. We do not trap data into the platform, and you can always export lists of studies. You can export screening decisions. You can export tags all in one click. All you need to do is filter Study Inspector to the studies that you care about and hit download, and it'll give you a spreadsheet of the data that you've collected. So we never trap data, and we want to set you up for success in whatever output you need to be sending or analyzing. On top of that, we talked a little bit about organizational settings, but we also want to make collaboration very easy. So we have comment features at any step in the software. You can comment to a member of your team. They'll be directed to that exact portion of the software you're working in, and they can answer your questions. You can also add people individually or in bulk. So if I just wanted to add my entire team, I can come in, I can add an organization, and then add my entire team to this nest in one click. And that will also give me access to my organization's template hierarchies, template protocols, template exclusion reasons, so that I can use that institutional capital, that intellectual capital, let's say, to build my review without having to do it from scratch. In addition, just a couple of more pieces that we always like to identify. You are able to make written outputs from this. You can use our manuscript editor. The main benefit of doing so is that you can create updatable tables. So if I simply select the columns that I want in a table, like let's say that I wanted a table of studies, and I wanted to add the column for types of hemorrhage reported, I can create this table, I can add it to my manuscript, and then as soon as any new study is brought into my review, it will be added to this table. If I change my tagging on this study, the tagging on this table will change. So say goodbye to manually editing every single cell of an Excel sheet or of a Word table. You can simply generate the table that you want and keep it ready for export into Microsoft Word to make your written outputs from your review, or even draft your publishable systematic review. And lastly, before I throw it back to you, Jeff, I was a little bit remiss in not showing this is all protocol-driven research. You can see here that this is an academic review that we completed with some Stanford neurosurgeons, but you should be using protocol-driven research to build all of this out. But the important piece within the software is to reflect those research questions in your search strategy, in your screening exclusion reasons, and in your tagging hierarchy. And so matching what is in your protocol to what you build into the software will set you up for success, and we will be able to track every single step. So making your Prisma charts, tracking every tag that you add so you are always able to revise them, and also tracking the data that are extracted and the heterogeneity, the bias related to each of them. So all of that is built into the software to try to reflect that protocol-driven research as effectively as possible in your outputs. And with that, Jeff, I think I've covered everything that I was looking forward to, and I'll throw it back to you.
Yeah, I would just like to add too, of course, that there's an element maybe of concern for some people whenever they see this and they're going, okay, so you've got all this AI in the back end helping, and it's shareable, right? I just want to take a moment to assure you that you own your data. And Kevin, you alluded to this when you were talking about we don't hold your data hostage, right? You can always export it at any stage, and even if you stop paying in the future for our software, you're always going to have access to your data. You're going to be able to pull that out of our system and use it for something else, right? So that's your intellectual property, and that is what businesses are built on, right? You can certainly go off and sell the outputs, and they are yours, right?
Can I do a speed round on these questions, Jeff? I see a lot of good ones.
Yeah, let me just throw stuff on the screen for you, and we'll see what can happen here. So let's do this one.
Yes.
First here, are there any institutional subscription plans?
Contact us. Yes, but we don't allow you to sign up online because we want to give you a custom discount. So contact us at sales@nested-knowledge.com, and we are happy to, basically all we need is the number of members of your organization, whether it's academic, and we'll make it happen.
Yep.
Collaborators. Your first Nest, no matter whether you were invited to it, whether you started it, is free. So your first project as a collaborator is free, but your second project would need a paid account. Next one. Oh, yes. Export is
just
great.
Some praise.
Yeah,
why not?
Thanks for the comment. Yeah, there are lots of great export features in there. We didn't even cover all of them. You can export bibliographic data. You can export meta-analytical data. There is an academic discount. So the academic subscription is $20. We actually charge a lot more for the corporate side, but we also we understand like international students and so on. So if you want a discounted student subscription even beyond the academic, that's a contact sales@nested-knowledge.com. More, Jeff. Okay. Now we're getting into content, and we have about a minute, so I'll go fast.
Yeah.
No, you can extract data from any study. So if you have prevalent studies, non-interventional studies, the only thing you need to be careful of is if you are extracting studies that do not report clinical outcomes, so intervention with outcome, the meta-analytical automations won't necessarily be relevant. We still offer options on how to extract prevalent studies and non-interventional studies, but they just won't generate those automated visuals. So yes.
Flexible structure. I just want to take a moment to plug our collaboration based on this question with GCES today, and maybe let the two doctors who are joining us kindly on this call to talk a little bit about that as well. But can we share a video on YouTube about all these steps so that we may learn from our free time? Thanks. Yes, we have tutorials and help articles on our website, on our YouTube channel. So if you're not on our YouTube channel today, just go to YouTube and search for Nested Knowledge, and you will find a whole bunch of tutorials that way. We also have a course though that Kevin did, and so maybe I'm going to try to
that is an entirely free course on how to do a systematic review. And also, I totally did not mention this in the demo, so it's a good question. If you want those YouTube videos and instructions on any of the steps that I've shown, you don't need to remember it from today. When you're doing a review, if I'm on the search stage and I have a question, I can just click the question mark in the upper right-hand corner. It's going to take me to a page that has a YouTube video on how to conduct a search and both text and visual instructions on everything that I've presented today. So if you're ever stuck, you can always contact support, but you can also always go to the question mark in the upper right-hand corner. That's going to bring you to our documentation that includes our YouTube videos, and it's going to be scoped to the step that you are on. But yes, Jeff, back to you.
Yeah, I just want to let Dr. Arvind. He has a few questions. Dr. Arvind, you still with us? He just said in the chat if he's not with us.
Thank you, Jeff.
Oh, go ahead. Go ahead.
Yeah, am I audible? Yes. So I was just wondering whether we can add, I mean, since you showed us that there's only one Cochrane risk of bias assessment tool, and when we are talking about multiple studies, can we add more risk of bias assessments that are coming up these days, JBI and even NHLBIs? So can we add those within the system?
Yes. So we currently support Cochrane, Sign, the Newcastle, Ottawa, and the JBI tools, but it's also very easy for us to add them. So if you have a risk of bias tool that you use commonly, just send it to us, like just a link to their questions, and we will build them into the software. And I also saw your question about tracking, and I didn't want to go into this too much because it's much more on the administrative side, but we have very intensive tracking. We track every person's screening, every person's tagging. If you want to see CAPA scores, individual CAPA scores within the software, there's an activity page where you can go in and view that. I think we'll save that for a later demo because I want to get to more questions before we finish it off. But yes, we have tracking. All you need to do is view the screening activity or the tagging activity of an individual within the software. But more questions?
Yeah, I think we honestly, Kevin, I think we should wrap it there. We have a lot more questions to answer, and of course, if you have questions for us, we're always happy to answer your questions. We have to end the live stream somewhere, right? So feel free to email us. I'm just going to throw that quickly back up on the screen here. Our email is contact@nested-knowledge.com. And you can just email us with whatever questions you have. I also just want to plug one more time the good folks over at GCES. You can scan this QR code to find out a little bit more about them. But yeah, we're doing this webinar today because they asked us to, and we were excited to at the prospect. So thank you so much for your time, and feel free to reach out with any questions. Any final words? Jade? No? Okay, I'm going to end the stream there. Okay.
The QR code for the sign-up page for Nested Knowledge in the top corner. Can't forget that either.
You can't forget it. You can't forget it. Okay, thanks.
Okay.
Let's talk soon. All
Thanks,
right.
everyone.